Hello Year 10 and welcome to the green penning of the B1 distance learning questions. As you know, we're at the end of B7 now. We've only got B5 to go, so we're back revising for paper 1. So we've got these distance learning questions. As I said, two really difficult questions. So long as you've given it your best go, don't beat yourself up if you found it difficult. There was some information drawing here from the topic B2 as well, which we'll be looking at in the week after half term. And the most important thing that you can do is understand the questions having gone through this video. Okay, so get your green pen done, and I'll be asking for some evidence of these questions in a few weeks' time. So we've got, after a meal rich in carbohydrates, the concentration of glucose in the small intestine changes. The table below shows the concentration of glucose at different distances along the small intestine. And then we've got the distance along the small intestine in centimetres and the concentration of glucose in moles per decimetre cubed. And the first question says, at what distance along the small intestine is the glucose concentration highest? Okay, so we've got the highest glucose concentration just here, and that is 300 centimetres along the small intestine. Straightforward for 1 mark. Use the data in the table to plot a bar chart on the graph below. Okay, so we've told us it has to be a bar chart. Even though we've got numerical data here, we don't have a lot of data. So I think that's why they've gone for a bar chart rather than a scatter graph with a line of best fit. It says you've got to label the y-axis, and that doesn't mean label it with the phrase y-axis. It means give it a title and choose a suitable scale. We can see we've got 4 marks down here for this. Okay, so the y-axis is concentration of glucose in moles per decimetre cubed. I planned it out. I thought to myself, okay, well, my biggest value is 500. So if I think 100, 200, 300, 400, 500, perfect, fits in nicely. Couldn't go any bigger and wouldn't want to go any smaller. So that's given me another mark. And then I've got my plotting of my bars. And you can see here, when we've got a bar chart, we put the value in the middle. We don't put it under a dash like I have over here. Okay, we put it in the middle as a label for that bar. And you get 2 marks for drawing those 3 bars correctly. Okay, quite easy numbers, quite straightforward to work with. Let's have a look at the second page. Okay. Look at the graph. Describe how the concentration changes as distance increases along the small intestine. So we've got this initial, if I go back to the graph, we've got this initial rise and then fall until we get to no glucose whatsoever, which is important, and we'll come back to that in a bit. So we get 1 mark for saying the concentration of glucose increases, okay, and then like a numeric value, okay, so we're referring to the data, so it increases between 100 centimetres and 300 centimetres, or you could say from 50 to 500 moles per decimetre cubed, so 1 mark. And then another mark for saying it then decreases between 300 centimetres and 700 centimetres, or from 500 to 0 moles per decimetre cubed. Okay, so you can use the distance or you can use the concentration of glucose. Either one works for your description. So we've got an increase and then we've got a decrease. If there's no numeric value there, then it's just 1 mark. Okay. Question D. Explain why the concentration of glucose in the small intestine changes between 100 and 300. So what happens in the small intestine? This is where we're bringing our mind back to B2. Okay, so we know that in the small intestine, we've got the amylase, which is an enzyme in the carbohydrates family, and that is going to digest that big starch into sugar. Okay, so carbohydrates, for example, starch, have been broken down, or you could even say digested into sugar, or you could say glucose, 1 mark. And then they are digested by the enzyme carbohydrase, or an example of a carbohydrase is amylase. So 1 mark for saying that the starch has been digested, and 1 mark for saying what does the digesting, and it's the enzyme carbohydrase. Okay, hopefully, even though you're thinking, okay, that's a bit more B2 than it is B1, hopefully that makes sense as what's happening in the small intestine. They do cross over a lot of these questions between different units. Okay, last part of this question then. Explain why the concentration of glucose in the small intestine changes between 300 and 700. So between 300 and 700, if we go back to our graph, we can see that's where we've got the decline. Okay, so where is that glucose going? Because it's being broken down from, well, the starch has been broken down to create the glucose. That created the high amount of glucose at 300 centimetres, but then where's it going? Okay, so key word, the sugars are absorbed. You can see I've underlined it there. So 1 mark for saying glucose or the sugar is absorbed through the small intestine. Remember, it's through the villi, and that is a link from B1 and B2 into the blood. Now, it's important that we say where it's going. It's not just escaping from the small intestine, it's actually ending up in the blood. Okay, so that's 1 mark. When concentrations of glucose are high in the small intestine, the movement is by diffusion. So we talked about this, I think we talked about it in this topic and in B2, where we said, okay, so we've got all of this sugar that's come from that digested starch in the small intestine. So it's obviously at really high concentration at first. That then diffuses across into the blood and we reach this equilibrium. Now, equilibrium means we've got the same concentration in the small intestine and in the blood. So how do we get that last bit of glucose across? It's by active transport. Okay, so we start off with diffusion. So when the concentration of glucose is high in the small intestine, the movement is by diffusion. As the concentration decreases, active transport moves the remaining sugar across. So we've got potentially 4 marks here, I think. I think we've got a mark for saying absorbed, we've got a mark for saying into the blood, we've got a mark for saying something about how it starts off as diffusion, and then to get the last remaining sugar across, active transport. So 4 possible marks, you only needed 3. Okay, hopefully you can green pen that bit and that all makes sense. So that's the end of the first question. Let's look at the second question, which was really quite hard. Okay, so this question I think came up a couple of years ago. It was on the triple paper, the triple higher paper. So, you know, just to give you an idea of how complicated this question is. One thing that they haven't told us that I think we need to think about is these have got to be boiled eggs, because if you're going to put them in acid and dissolve the shell, if it wasn't a boiled egg, it would just, you know, it would be runny and liquid. So it's a boiled egg. Sorry, I just knocked the stand there. They've used chicken eggs, and it says this is the method used. Place 5 eggs in acid for 24 hours to dissolve the eggshell. Now that's the only thing the acid is doing. It's basically peeling the egg for you, because if you peeled an egg, you can imagine that, you know, we've all done it. Sometimes it peels off really nicely. Sometimes, you know, you pull away bits of the egg white at the same time. So I think the reason why they've dissolved the shell is so that it just removes the shell and the boiled egg is intact underneath. Okay, so measure and record the mass of each egg. So we've dissolved the shell gone, acid gone, not part of it anymore. Part 3, place each egg into a separate beaker containing 200 centimetres cubed of distilled water. After 20 minutes, remove the eggs from the beakers and dry them gently with a paper towel. And we talked about that in the potato osmosis practical, getting rid of the excess water so it's not part of our measurement, and then measure and record the mass of each egg. So they've been in water, so we're expecting to either have water loss or water gain, okay, because there's going to be some movement of water by osmosis. If there's no movement, the experiment is pointless. Okay, let's look at our data. Now, I've done quite a lot with the data because I was thinking back to what I already know. Remember, they want us to apply our thoughts to new situations. So I was thinking, right, what did we do in the practical? Well, we had the start mass, we had the end mass. We then worked out the change in mass by having this number at the end minus this number at the beginning. That gave us the change in mass. If we divide that by the start mass, you can see I've worked out one here, and times it by 100, that gives us the percentage increase. Now, because you can see they all started out with different masses, we have to do a percentage increase in order to fairly compare our results. So I've shown you kind of one completely worked out, 77.0 minus 73.5 gives us an increase of 3.5 grams, and then we divide that by 73.5 because that's the original mass of the egg, times it by 100, and we get a 4.76% rise in the mass of the egg. Okay, so water must have gone into that egg. In fact, water's gone into all of the eggs because we've all got positive percentage increases. Okay, we've got no decreases there, which should be represented by a minus here. Then I worked out the difference here. I wrote it out slightly shorter this time. So I found out that the difference was 3.6, divided it by the start and times it by 100, and there we go again. And I've done that for all of them. Now, I knew that I needed to do that to be able to fairly compare my results. You might not trigger in your mind, that's what I need to do in order to access this question, but later on, in the question it asks you to do one of these calculations, so then it would jog your mind. Okay, so I just did it because I thought that's going to help me in the question, that's going to be useful to help me answer the rest of the questions. So another student suggested that the result for egg 4 was anomalous. Okay, that means it doesn't fit the pattern. Do you agree with the student? Now, because it's only 1 mark, we're not going to get a mark for saying whether we agree or not, but I've gone with yes, and you can see we've got over 4%, nearly 5%, 5%, over 4, only a 2% rise, so less than half of the others, and then a 4% rise again, okay? So the mass change is lower than the others, okay? So you can look at it as a percentage and say the percentage change is far smaller, doesn't fit the pattern, or you can say, if you just look at the difference between the two masses, you can say all of them gained over 3 grams except for number 4, which only gained 1.5 grams. But again, even that's a 1 marker, look how I've put data in my answer. Biology loves you to put data in, okay? So many of the mark schemes show that if you put data in, you get more marks, okay? So the mass change is lower than the others. They all gained over 3 grams except number 4, which only gained 1.5 grams. So something went wrong with that experiment. We don't know what caused that, but we could think about that if they asked us to, but I don't think they're going to. Okay, moving on. So now it said, calculate the percentage change in mass of egg 3. So we've used the table. We had 75.5 minus 72.4 gives us 3.3, so that's the mass increase. Divide that by the original mass, 72.4, and then for it to be a percentage change, we times it by 100, and we've got an answer there. And biology isn't like chemistry and physics, where it's all about, or currently it's all about recording our numbers to so many significant figures. They're quite flexible. I went with 4.56%, but they will accept 4.6 or equally 4.558. So any of those would be fine for that answer. Okay, always good to show your working, because if something goes wrong and you've not shown your working, that's it, you've lost everything. It's amazing how many people make they get all of the working done correctly and then they don't read the calculator properly, or they round it incorrectly, and then they've lost those marks. Okay, next one, part C. Explain why the masses of the eggs increased. So I've already mentioned about osmosis. So we've got a 3 marker. First mark, water movement in to the egg by osmosis. So we've had water go in, that's what made it heavier. And how has that happened? It's happened by osmosis, 1 mark. And osmosis is from an area of high concentration of water, which is in the beaker, to an area of low concentration of water in the egg. Now, notice how I say of water, because it's the water that's the important thing. There could be salt or sugar or something in the egg that is causing it to have a different concentration of water than in the beaker. Okay, so it's a higher concentration of water in the beaker, low concentration in the egg. Therefore, that's why it moved in. And osmosis always involves a partially permeable membrane. Throw it in wherever you see osmosis and you will get yourself an extra mark. Okay, moving on. Question D. Explain how the student could modify, so what changes could they make? We don't need to write a whole method, it's only 3 marks, but how could they modify the investigation to determine the concentration of the solution inside each egg? Now this is what we did in class. When we had all those different sugar solutions at different concentrations, and then we plotted a line of best fit that looks like it does on the osmosis video. If you have a look in the playlist for B1 and you take a look at me doing that practical, we've got this kind of graph like this with the x-axis, and we've got a line going down like this. Okay, where that line crosses the x-axis, that concentration where it crosses is the concentration of sugar or salt or whatever it is that we're investigating inside that food. So what could we do to modify this method to work out the concentration of whatever it is inside the egg, whether it's salt or sugar? They haven't been clear. So we've got to use 5 or more. Now the mark scheme says you've got to have at least 4. So 5 is, is a good number. That's what it says on the mark scheme. So use 5 or more different concentrations of either sugar or salt solution, whatever it is we're using. So that's 1 mark. We're then going to plot the percentage change. So we'll do all the percentage change the same, so we don't need to tell them what we're doing, but we'll plot the percentage change in mass onto a graph. And like I said, it will give you, I'll just draw a little sketch now so you're clear on what I'm talking about. Here's our percentage change in mass. Here's our x-axis with all of our different sugar concentrations, and we'll have a line of best fit that looks something like this. Okay, and it's that point there, look at me with all the pens, that point there, whatever that concentration is along there, is the concentration of the salt or sugar solution in that egg. Okay, so where the line of best fit crosses the x-axis with no change in percentage mass is the concentration inside the egg. So that's going back to our original experiment in school. Now, we really hammer home at the time, just because we're looking at potatoes in school, they can look at anything. And that's why they've thrown us in at the deep end by talking about eggs. We didn't know that we could do this, but we've kind of visited the original experiment in this question. Okay, moving on to the last page. Okay. Chicken eggshells contain calcium. So we've now gone back to the shell, even though we destroyed it with the acid earlier. We're now talking about the shell again. Calcium ions are moved from the shell into the cytoplasm of the egg. Okay, so there's calcium in the shell, and the ions move from the shell into the cytoplasm of the egg. Okay, fine. Table 2 shows information about the concentration of calcium ions. So we can see in the eggshell, there is a low concentration, 0.6. Arbitrary units is just, it's not a unit, it's just like a scale, if you like. Like if I said, how happy are you on a scale of 1 to 10, you'd give me your answer as whatever it would be, hopefully 10, and it would be in arbitrary units. It's just relative to the other numbers on the scale. So 0.6, which is a low concentration compared to in the cytoplasm, which is 2.1 arbitrary units. So we're going from a low concentration to a high concentration, and we know that one should be alarm bells ringing, active transport. So explain how calcium ions are moved from the shell into the cytoplasm of the egg. And it says calcium ions are moved from an area of low concentration to high concentration, 1 mark, defining it. What's the process called? Active transport, 2nd mark. And how does that occur? It needs energy. And that energy comes from the mitochondria, it comes from respiration. Okay, now I know that I've given you some really challenging questions. It was my intention to kind of stretch your knowledge, push those people who are really, trying to get those good grades and not just sit on our laurels and just kind of glance over the stuff. I really wanted to push our brains today. So well done for that. Hopefully you understood it. Sorry it took a bit longer than I anticipated, but hopefully you've got some really good learning from that and good understanding of exam questions. Okay, have a good day. Thank you. Bye.